Hey guys, we're going to go over the lesson today about cellular energies. So think about, what do we need energy for? There's lots of things. We need energy for synthesis, for building biomolecules or macromolecules. We need it for reproduction of organisms and for reproduction of cells. We need it for movement. We need it for active transport. We need that energy to be able to move things across that cell membrane. And we need it for temperature regulation so that we're not too hot or too cold. Okay, and so this gets at our idea of living systems needing energy, which we've talked about in the past. We've talked about the law of conservation of energy, which is that energy cannot be created or destroyed. It can only be transformed or changed into something new. And so living systems need a constant input of energy so that they can reproduce and maintain organization and all of those things. So think about, where do they get this energy from? And they get it from the chemical bonds that are stored inside of those chemical bonds, usually in glucose, or we'll use ATP for energy. And this is our first law of thermodynamics, this law of conservation of energy. Okay, I'm going to move it over here now. So where do we get energy from? Where do they get energy from? There's several different sources. We could have energy coming from the food we eat, or the sun, or there are some exceptions, such as like deep sea vents here, where there are bacteria and things growing down there. Okay, so our things using the sun are going to be autotrophs. The things using usually eating either other autotrophs or heterotrophs will be called heterotrophs. And then our ones that use deep sea vents are our chemotrophs, which are pretty rare. Okay, and what organelles are involved with this energy? We've talked in the past. This is our mitochondria and our chloroplast. So we want to think about what energy processes are taking place in these two organelles. And so in our mitochondria, that's cellular respiration, and we will talk more about that in a week or so. And our energy process for chloroplast is going to be photosynthesis. So cellular respiration, what's that? That is turning our food, usually glucose, into ATP, and our photosynthesis is turning sunlight into carbohydrates, which would be glucose or food. So why do we use ATP? So just like we talked about, energy can be stored in bonds. We use ATP because it has bonds that can be easily broken, so we can have a quick energy that is being stored in those bonds of the ATP. Okay, we also have a thing that's related to our idea of energy, and that is entropy. So entropy is looking at our disorder or chaos in a system. So any closed system will tend towards a maximum state of entropy. So it's going to go towards disorder unless we have some energy input into it. So our life, though, is highly ordered, not disordered. So therefore, it doesn't break this rule. In order to get this ordered nature of our living creatures, that is because organisms use energy and are constantly inputting energy into their system in order to decrease that disorder or entropy. Okay, so notice here that it's a closed system will go towards disorder. Our cells and our bodies are not a closed system. They're not at equilibrium because we need that constant input of energy. This is our second law of thermodynamics, the idea of entropy. Okay, and then this is related to our idea of Gibbs free energy, or just free energy sometimes, and this is the amount of useful energy. So this is our Gibbs free energy, and here we have enthalpy, temperature, and entropy. So these are all things, you do not need to know this equation, but these are all things that are related to how useful energy is, our stored energy, our temperature, our disorder or entropy of a system. So we can use this free energy to look at our different types of reactions. And we can have two different types. We can have exergonic reactions, which are shown here, where we have a lot of energy, free energy in our reactants, and less in our products. Okay, so if we're looking at that change in free energy, we would do our products energy minus the reactants energy, and we would get an overall negative change. Okay, it's because it's going down. So we would have a negative change in free energy. And usually we think negative, we don't want negative, but in this case we do. That negative means that this reaction will happen spontaneously, which we want to happen. We want reactions happening spontaneously in our cells. Otherwise, they would not be occurring at all. Okay, so here energy is going to be exiting. That's why it's called exergonic. Okay, so you can think about it going down. Similar to our concentration gradients, we're going from high energy to low energy, it will happen naturally. It will happen on its own spontaneously. Okay, endergonic reactions are going to be the opposite. Here we have less energy in our reactants and more in our products, which when we do our products minus our reactants, we will have a positive free energy change, which means that they will not be spontaneous, which means it's not going to happen on its own. We need energy to be inputted. So energy will enter, so endergonic enter. Here is like going up a hill. It requires energy going from low energy to high energy. We need to use energy to push it up. And so biological systems use both endergonic and exergonic reactions. Okay, so our exergonic reactions will provide that energy, that free energy that's necessary for our endergonics. We put those two together. So like here we have our exergonic reaction that is having energy exit that can then be used to go up into our endergonic reaction that requires energy. Okay, and so this gets at our idea of our catabolic pathways. So catabolic pathways are, or catabolic reactions, are examples of exergonic reactions. So these are where we are taking and we are breaking down molecules. So breaking it into smaller molecules, like digestive enzymes breaking down food. This is going to release energy. Releasing, it's exiting exergonic reaction. Okay, so here we have a large molecule. We're breaking it down into its parts or monomers, if it were like a carbohydrate or a lipid, and we were releasing energy. That is a catabolic reaction. Okay, so here we have C is not for cookie. C is for catabolism. So our catabolic reactions, breaking that down for energy. Our endergonic reactions in biological systems are like our anabolic pathways. This is the opposite of catabolic in that instead of breaking down, we are now building. So we are using energy, again, why it's endergonic, energy is going in, and we're using it to build more complex molecules. So an example would be amino acids forming a protein. Here we have our monomers being formed to make a polymer. Okay, so here we have an example of an anabolic process using proteins to create proteins. Okay, and both of these terms, anabolic and catabolic reactions, are related to this idea of metabolism, which you've probably heard before. Okay, so metabolism is the totality of an organism's chemical reactions. It's all of the chemical reactions, so that's the endergonic and the exergonic reactions. So they're going to be using the energy resources in the cell. So it's all of them put together. So our catabolic reaction is breaking down the molecules, and then our anabolic reactions are building different molecules. So everything that's happening in our cells is breaking down something, building it into something else. And this gets at our idea of having our endergonic and exergonic reactions coming together, and we call that energy coupling. Okay, so we are linking our endergonic and exergonic reactions so that it's more efficient, and we are able to have the energy required for those endergonic reactions. So overall, we'll have a negative delta G because the negative delta G we have with an exergonic reaction will outweigh the positive from the endergonic reaction, making it together overall negative. Okay, so again, with our example of this ball, the energy from going down can then be used to go up the next hill. So this is coupling these two together. Okay, so now we want to think about how does the cell capture and release this energy? So it needs some sort of currency to help it to pass this energy around. And it needs to be a short-term energy molecule, and it's going to be ATP that we use, adenosine triphosphate, ATP. So what is ATP? So ATP is actually a modified nucleotide. So it's an adenine. That's where this adenosine comes from. It's a modified adenine. It's got a ribose, and it's also got some phosphate groups. Usually we denote that as PI. I stands for inorganic phosphate. So we have 3, which is why we get the triphosphate. So we add phosphates, that's endergonic. The opposite is true. Removing those phosphates would be exergonic. So you can also think about ATP kind of as a battery when it has all of its phosphates here. It's a stored battery. As soon as we remove that one of those phosphates, it doesn't have energy anymore because that energy is being released, and we can use that energy in some other reaction. Okay, so all of these reactions would really happen very, very slowly. But we don't want that to happen. So we are going to have catalysts in our cells that are able to help with this without altering the process. Okay, so that's our enzyme is our biological catalyst, which we've talked about in the past. So we have our enzyme and our substrate and active sites. And again, what they're doing is they're lowering this activation energy. So they're making it so this reaction can happen a lot faster and more efficiently. Okay, and so our enzyme has an active site that is specific to a substrate, so it's only going to be catalyzing certain reactions, so we can control what is being catalyzed and what is not. So the enzyme will bind, or the substrate will bind to its active site, and it forms what's called an enzyme substrate complex. So it makes this kind of complex with the enzyme and the substrate bound, and this is also called the transition state. And the enzyme is stabilizing this transition state for our molecule, which then makes it so that the product can be formed more easily. So this active site is where it's being bound to, and that energy has that enzyme adds the additional energy that will speed up the reaction. Okay, in the past we've talked about how the shape determines the function. So the shape of our enzyme will determine the function of what it is doing. But sometimes enzymes need a little help getting the right shape so that they can have the proper function. So there's some things that can help with this. Cofactors and coenzymes are going to help our enzyme make the correct shape. So cofactors and coenzymes have a similar function, but they're just made out of different things. Cofactors will be made out of minerals such as zinc, iron, or copper, and coenzymes would be like vitamins that are involved. So they're going to help make the correct shape for our enzyme. Okay, and all of these enzymes and energy that's happening is going to be happening in pathways, and these pathways are going to be sequential, so happening in a sequence. So we have a starting molecule, and then we can have an enzyme that catalyzes that reaction to turn it into something else, and then we can have that product being used as a reactant in our next part of our pathway, and a different enzyme can catalyze it to be made into our next one, and so on and so forth, where we have enzymes helping to make our product, and it changes along the way. So it's a pathway. Okay, and we can control this pathway so that we have efficient use of our energy. Okay, so we can regulate it with different ways. So one way we can regulate the pathway is to have the genes that encode the enzyme. So if we synthesize less enzyme, we make less enzyme, we're going to therefore catalyze fewer reactions. So if we're making too much of something, just make less enzyme, and then it will not be catalyzing those reactions as quickly. Opposite is also true. If we want to have more of something, we can make more of that enzyme to speed up the reaction. We can also have allosteric regulation, which we talked about when we talked about enzymes in the past. So allosteric is dealing with a spot other than the active site. So we can have things that are going to regulate our enzymes. So we can actually have activators that will bind allosterically but actually activate. So they will stabilize that active site. Or we can have inhibitors, which we talked about in the past, which would stabilize the inactive form, making our enzyme have a conformational change and not be able to function properly. We can also have cooperativity, where one substrate triggers a shape change in another active site. So very similar. Oops, I don't know what I clicked on. Okay, and then so with this pathway and our enzymes, we also have something called feedback inhibition that can help us to regulate our pathways. So this is where the end product can shut down. Sometimes we can also have where it either shuts down or starts a pathway by binding to an allosteric site of the enzyme. This is preventing waste usually. So if we've made a bunch of a product, we don't want to just keep making more of it if it's not needed. So we can go back and turn off the pathway so that we don't make excess amount. So here we have an enzyme that is catalyzing an end product, and then that end product can then serve also as an inhibitor of our earlier enzyme so that we don't continue to make a bunch of our product. Okay, so that would be our negative feedback, where we have our end product inhibiting, but we can also have a positive feedback where we're just making more and more because that end product is being used to signal and activate our enzyme. Okay, so in class, that is all of the slides. We talked about doing the ATP POGL, so that's due for next time. There's some resources, like there's a video on Life Requires Free Energy that you can watch, those textbook chapters, the focus questions. Don't forget your Unit 2 progress check. All those things will be helpful. Bye, guys.